Welcome back to the advanced module. I hope you're excited. I see new hairs here. Short guy. Very cool, very cool. I hope you enjoyed your week of vacation and had a great time, especially the people that are not from Brazil. I hope you enjoyed a little bit. So we will start this advanced module, and it's my big pleasure to introduce to you Deborah Toiber. Deborah, we usually call people by their first name. I cannot call you Toiber, it's too weird for me. Debbie is okay. Great, we like nicknames. So Debbie is a professor, is a molecular biologist with a PhD from the Hebrew University of Jerusalem, followed by a postdoc at Harvard University, correct? Currently, she's a professor at the Ben-Gurion University of Negev in Israel. Is it correct? Negev. No, we tried our best. So her research focuses on molecular mechanisms behind aging and changes that happen as we age. Work especially in the DNA damage accumulation and the risk of developing age-related diseases. Okay, so we will start like in a good mood and a good and we hope you all enjoy this new module and help me to welcome Debbie.
Okay, thank you. Okay, thank you very much for the welcome, and I'm very happy to be here. Today will be, I understood that you come from different backgrounds, so today it will be a very basic class to remind you all that you were supposed to study in a first degree in an hour and a half. And then from tomorrow we will start on genetics and epigenetics and the important things. So we will have about the cell cycle and about stem cells and reprogramming, about DNA repair and aging, because it's influenced by genetics and epigenetics. So today be patient, those of you that really know biology. It may be very basic for you, and those of you that come from different backgrounds, it may give you the opportunity to catch up on what we are doing. I don't know how to switch back to the presentation. Let me. Okay. No, not that. How come? There it is. Okay. Also, I tend to speak really, really fast, and I don't get upset if you just raise your hand and tell me, go slower. I speak Spanish, in case some of you prefer also Spanish. And that's it, so I hope it will be clear for everyone, but don't hesitate to just raise your hand and say, please go slower, okay? So today is very, very basic. We want to study biology and do research in biology. So we started with a science that was very observational. People just look at the nature and describe it. But nowadays, we need many, many different tools from physics and computational biology and chemistry and many, many things to do research in the highest level. So of course, we need programs like this program to advance biology. And we try to answer through these methods all the biological questions that we can think of, from how to evolve to be where we are today, and what do we do when we get sick, how to feed the world, how to not destroy the world, and all of these big questions is what we hope you in the future will manage to help us solve and move science forward. So these are just a very nice short movie about life on Earth. Let me see if it works.
Life, with an estimated 3.8 billion years of existence and evolution on Earth, is an odd thing indeed. So how did we get here? And what would the Earth's 4.5 billion year timeline look like compressed into a normal day of 24 hours? The story begins early in the morning, around 4 a.m., with the rise of the first simple single-celled organisms. However, these organisms are destined to spend quite some time alone. At 1 p.m., a simple cell engulfs another, creating a symbiotic relationship and the first eukaryotic cells, or cells with internal organs. At 6:30 p.m., these cells begin to form colonies, and thus the first multicellular life develops. But it isn't until 8:30 p.m. that sea plants appear, and 20 minutes later we suddenly see animal life erupt in an explosion of inventiveness. Jellyfish, the first true vertebrates, trilobites. Incredible diversification begins to take place, and just before 10 p.m., plants begin to appear on land, followed by land mammals. By 1024, the Earth is covered in carboniferous forest, and the first winged insects appear. Many reptiles dominate the land with less than an hour left, but a mass extinction event allows dinosaurs to storm the scene and remain until 11:41 p.m., when they too suddenly vanish. And so begins the age of mammals. In the last few minutes, apes split from the old world monkeys, and with only 1 minute and 17 seconds left before midnight, humans emerge. All of the recorded human history fits within a few seconds, an individual life lasting barely an instant. All a culmination of 3.8 billion years of life. This ASAP Science video was made possible by the life with.
Okay, so this is just to give us a scale for how long during all this Earth existence we have been existing. And as you see, mammals and all of us have a very few seconds in the history of the world. And mainly we have many different unicellular systems. And we work as well in biology, not only in time, but also in different scales, as you can see here from the first proteins that can already have a very strong reaction. For example, prion disease is caused by misfolded proteins, to adult female or whales or the very big organisms that we know. So we work in all of the different scales, depending on what we are focusing on. If you are using cryo-EM, you can get to atom resolution. And if you are doing ecology, you will get to a population resolution. So all of these different scales will help us understand better what is happening in the biological world. So the basic unit of life is cell. And again, many years ago, they tried to come up with what is the basic unit and what is life. So it has to be organized, it has to use energy, it maintains internal environment, it reproduces with molecular heredity, because DNA or RNA it can be, depending on some of the definitions. Some people consider virus to be alive, and some people don't consider virus to be alive. And it's still a debate of what we mean by life, because many of these characteristics can also be nowadays in a computer model. It can be kind of alive. So it's difficult to raise what is actually alive, but mainly the use of energy and the changes in metabolic features are what make us alive. It grows and it evolves. So this unified theory of the cell came many years ago by Rudolf Virchow, that says that from a cellula comes the next cellula. So we don't have a spontaneous generations of cells. Always we have a previous cell that gives rise to the next cell. All the organisms are made up of cells. This is the basic unit. And we can have a unicellular organism or multicellular organisms, and they arise always from cell division. And we will have different kinds of cell division. Like from yeast, we can have an asymmetric cell division, and in other cells, symmetric cell division. And we will see also this already in development, that we have asymmetric cell division to give rise to different types of cells that can give us a full organism. So we have, again, this is very basic. It's just to remind everything about biology that we are supposed to know. So be patient, and I promise tomorrow will be much more interesting for those of you that is very basic. So we have prokaryotic cells. They don't have a nucleus. They are bacteria, archaea, and are the most simple ways of life. And we have the eukaryotic cells that already have an organized nucleus with genetic material that is contained in this nucleus, and it is also regulated very tightly. So we have different in the cell size and how this genetic material is organized. Usually as we progress and we have eukaryotes, we have histone protein likes to form the chromatin and to allow us to have a very important regulation. So mainly if we tell you today a yeast has almost the same amount of genes that a human, This is true, but what is different? And different is regulation, because we have the combinatorial power of using these same genes in different ways. So by adding regulatory layers, we can form many, many more functions. So most of the energy we spend in every cell in our body comes from regulation, and mainly from epigenetic regulation of the DNA. So of course we have genetic variants and we have many different things, but if you remember, each cell in our body has the same genetic material, but each one of the cells has a different function. How we attain this, and it's mainly through epigenetic regulation. And this will be the focus of our next classes. So this again is in a very short schematic way to show you all the cellular compartments, and then we'll go one by one, all these functions. So when we start our next class, you at least have all these Golgi and ER and different things really in your mind. Let me see.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment. Cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So what are organelles? Organelle means little organ. Organelles are the specialized parts of the cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the Endoplasmic Reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The Endoplasmic Reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the Endoplasmic Reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, The mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-closed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Okay, just to be sure, I know that these are very basic. I don't know how many of you are coming from non-biology. Raise your hand if you're coming from physics or something else. Okay, and those of you not coming from biology, what are your bachelor's degrees? Where? Computation? What? Mathematics. Okay. Physics. Okay. Mm-hmm. So I was told that this is very combined. I hope those of you that know biology don't get very bored, and I hope that we will get to more interesting things. So I'll try also to put you in the perspective of what is known today or not. So we will start with the cellular membrane. Although this sounds very simple and everybody knows that there is a membrane, no one really understands in evolution how it was formed, how the first membrane was formed. Because we are closing something, but it needs to be permeable. It needs to allow some things to go in, some things to get out. We never get a new membrane formed that is not coming from a membrane. So membranes, although it sounds like just some envelope, are very, very complicated, and we don't completely understand through many, many different theories in evolution how the first membranes were formed. There is active research on membranes and how they were formed. Usually, it gives the cell also the shape, and it allows the passage of different materials and prevent the passage of other materials. So they think that at the beginning we have this kind of micelles, like what is formed with the soap, right? And they were not very permeable. They have many things, but then things started to organize inside according to their biochemical properties. And nowadays, we are doing very active research on what is called liquid phase separation. So at the beginning, maybe they were not real membranes, but we have this liquid phase separation that is like putting oil would be already a membrane, but oil in water, we see it separates immediately. So also we have some RNA or some modifications such as pyrylation and some proteins that form these characteristics that separate them from water. So maybe the beginning were not real membranes, but this kind of liquid phase separation that allowed the formation of domains that later on they got more complicated and gave us the membranes. So we know that the membranes have this phospholipid structure, and that if we have more double branches, this prevents them from freezing. right? So that's why in many cases the cholesterol helps from the membranes completely freezing in low temperatures, right? When we freeze our cells in the lab, we always add some glycerol or some other molecules to prevent them from exploding because water expands when it's frozen. So cholesterol is one of those molecules, but if we have too much cholesterol, then we also start causing other type of damage. Or for example, we hear all the time that it's good for us to have omega-3 fatty acids that usually we take from fish that live deep under the sea. Why? Because they can live in very low temperatures. They have to adapt, and their membranes have more of these fatty acids that prevent the membrane from freezing. So these are some of the characteristics of the membrane that are important and can vary depending on the temperature the organism is used to live. So now we have a membrane, we need things to come across the membrane. So we have different kinds of transport that have to occur. We have simple diffusion, molecules that are small enough or they can cross these barriers that can just pass. We have passive diffusion that it depends on the concentration, and we have some channels that allow the passage of these molecules. And then we have to create energy to let some of the things pass, so we can have symport that one molecules come one way and the other way, and we have different kinds of transporters, some of which require energy, like ATP, to allow the passage of different molecules. So also this membrane is constantly needing energy to allow the proper function. And another thing that the membrane does is allow the cells eating. We can have phagocytosis, so we can internalize solid big particles inside the cell. And sometimes this endocytosis can also internalize pathogens. We can, by mistake, engulf a pathogen that uses the acidification of these vesicles to be activated. So we have some viruses, for example, that when they are internalized and the vesicles are trying to digest this as if it was food, they actually get activated and start the process to reproduce and kill our cells. So it works in a good way for us. This is how we eat at the cellular level, but also evolution has made some organisms to take advantage of this. We can also drink. Pinocytosis is like a more fluid-like kind of eating. And we have the receptor-mediated endocytosis, meaning that if you want to get in, you have to have the right signal for me to allow you to get in. So we have receptors that allow this internalization of specific things inside the cell. Another important characteristic of the membrane is the signal transduction. We want to get information from the outside to let the cell know what is happening, right? Because we work in a multicellular level. So for example, if we want to know if we just ate and we want to process the sugar, we want to release insulin and start the process of taking up the glucose and breaking it down. So we have receptors in the membrane that allow this signaling, like if it was an antenna that allows the passage of this signaling inside the cell. So as I was telling you, we have a receptor that can recognize a single molecule that activates intracellular mechanisms, like a domino effect. We throw the first tile, and then we can start the activation of all of the molecules to get the cellular response. So for example, we can have signals that are sent from the cell and received by the same cell. This is called Autocrine Signaling. So I send a message to myself as a reminder of what is happening, like a WhatsApp to yourself to remember something that needs to be done. Or we can send a cell from one cell to the other, but through gap junctions. These are the things that are connecting two cells that are neighbors. So we can also share some of the material, maybe sometimes the mRNA and some other things. We have the paracrine, that are signals that are secreted and then get to the target cells. And the endocrine, that they really have to go through the bloodstream to get to the next cell. So all of this can give us how a cell communicates with each other through different mechanisms, and mainly all this communication will have to pass through the cell membrane. So as I was telling you before, we have insulin that binds to the receptor, and then it gets one of the most used mechanisms in biology is phosphorylation. Why? Because it's very simple modification that can be done very fast it uses ATP as energy, and it makes a big modification in the protein. Phosphorylation is relative, a big modification, so it can change the interaction of the proteins that start the signaling, for example, to have the glucose receptors going to the membrane and start picking up glucose from the outside. So we start with a signal, we activate an internal mechanism, and then we allow the signal to respond and to adapt to the environment. So for example, if we have some people that eat too much glucose, then we are constantly activating this signal, and this leads to an adjustment in the cells that in the end of the day, they get exhausted and they start dying because they are hyperactivated. And this is one of the theories in the long term why we get type 2 diabetes, right? We are killing the cells by hyperactivating them with signals of glucose. So this signaling is not only to keep homeostasis, but if it's overused or used incorrectly, it can lead to disease, okay? And just also as a reminder, not all the signals are chemical or peptides. Of course, in the neurons, we have a lot of chemical molecules such as acetylcholine that activate receptors and send signals from one neuron to the other. But also we have the electric signal that is transmitted in the cell by changing the membrane potential, right? So this goes like a wave and get to the next cell. So we have various different types of signaling that can go from the membrane in different ways. Receptors, allowing things to go in, allowing things to go out, electric potential of the cell, and all of this is thanks to our membrane. So they have very important roles. Next, okay, we are inside the cell. We go to the genetic material. That will be the main focus of the following days as well. And at the beginning, when research started in biology, no one thought that DNA could be an interesting molecule. It looked very boring. It was repetitive ATGC compound, a chemical compound that no one imagined that could have information. They thought that proteins, because they have 20 different amino acids at least in several combinations, this was the genetic material. So they started doing many different experiments to try to find out which one was really the genetic material. And one of the first experiments, but this was not the decisive one, in around 1928, was that someone discovered some virus that was not very virulent. The mouse could live if it got infected by this virus. And there was an alternative one that looked rough and one that looked smooth in the microscope. And this smooth one will kill the mouse. But if it was heated, and the smooth one, then the mice would not be killed. And then the interesting thing happened is they mixed the heated smooth one with the rough one, and then the mouse died. Now this is very weird way to confirm that DNA is the genetic material, but what happened is the virus was degraded, but the DNA material was there. And virus could uptake this material, and when infected the cell, we will get the two genomes. And then we have the one that is pathogenic and will kill the cell. So this was not proof enough. But then came a very interesting experiment using this bacteriophage. So bacteriophage are these very tiny viruses that infect the cells, and they look like this aerospace ship. They stand on the membrane or the wall of the bacteria and then inject the DNA inside. So what they did is they labeled the proteins with a radioactive material in one of the cases, and in the other case they labeled the DNA with radioactive material. And they wanted to see what is happening. So they took, after this radioactive phage, and they took the cells that were infected and checked if the radioactive material was inside the cells, meaning in the pellet. All the bacteria, when we centrifuge them, they go to the bottom, and all the things that were not inside the cells stay outside. So all the proteins were left outside, and all the pellet has no protein radioactive label inside. In the second experiment, the DNA was labeled, and then all the radioactive material was inside, and all the proteins were outside. So this was a decisive moment or decisive experiment that showed that DNA is the genetic material. This is the one that gets inside the cell and managed to get all the reproduction of these bacteriophages, again to produce more bacteriophages. So this was the experiment, very elegant with the methods that were available at that time to show that really DNA is the genetic material. So we will focus in this lecture and also the next one in animal cells, mammalian cells, and less in plant and bacteria, because what we know about epigenetics is more relevant for mammalian cells. So we will go slowly through all the organelles, and we will start with the nucleus, but next class we will focus really on the nucleus and all the nuclear bodies that are inside. But in this case, we just give you I'll give you a reminder of the genetic material, how it looks like, and we have the DNA, that is DNA and RNA that are formed by these nucleotides, cytosine, guanine, adenine, and uracil, and they really look like sugar. You know, this is just simple sugar, and these are our nucleotides, and they are bonded through the sugar and phosphate bond, and RNA is single-stranded, but it can form bonds with itself and form different structures, and these structures are very important for also epigenetic regulation and also for regulation of RNA lifetime interactions and many other things. So RNA can form different loops, and these loops have information of how can they bind DNA or other proteins and give the regulation of if it will be expressed, it will form a complex that does something different. And we will see more about it tomorrow. And DNA, as you know, is a double helix that has a 3' and a 5', so it has some polarity, right? And it's linked together through hydrogen bonds, like the water, right? If we have a water molecule, what links the water together and it gives its properties is these hydrogen bonds. So these are the same kind of bonds that we have in our DNA. But if we have GC, we have stronger bond, we have 3. And if we have TA, we have 2 bonds. So usually when we want to keep some regions of the genome tight together, we put more GC-rich sequences because it's more difficult to open them. It requires more energy. So if we have a region that is GC-rich, we know that it's a region that requires extra energy, so maybe an helicase or something else to be open. An 80, it's simpler because we have 1 extra bond. It's okay? Great. Okay, so we have these bonds. And then we have, again, the DNA replication that we know is semi-conservative. So from one DNA, we get another DNA, but the original strand is divided between the 2 new molecules. So each molecule is copied on the basis of one of the strands, and then we get the second one. And this is also very important when we'll get to epigenetic modifications, for example, methylation on the genome. You know what methylation on the genome is? Most of you know? Okay, we will get to it tomorrow, but we have the genetic code. That is what we say, ATGC. This is the most common. Of course, we have some modifications that can happen. And this is the genetic code. The epigenetic code has 1 modification at the level of DNA, and many modifications that are on the chromatin itself. And one of these modifications is methyl that can be added into the DNA. But if it's in one side, when we replicate, how this information is divided, because sometimes it's not symmetric, right? So this will be one of the problems that we have when we are replicating to keep the epigenetic code, and we'll get to this tomorrow when we go to the nucleus. So we are adding we open the DNA, and then if we had a T, we add a A, we have a C, we add a G, we have a A, we add a T, and in this way, we replicate the information exactly as it was, right? And then we get this semi-conservative mechanism that we have two new DNA molecules, but each one has an old one within. So DNA has directionality. We have like a phrase. It starts with this capital letter and ends with a dot. We have this 5' and 3' that are different, and not all of the enzymes know how to work in both directions. So this gives us a problem when we try to replicate our DNA. So what is happening is that if we go from 5' to 3', we can do it with no problem. We have enzymes that can do this without no problem. It's energetically very easy to do that. But when we have the second strand, we have to go again from 5' to 3', but we are on the other side. So what cells do is divide it in small fragments, put a little primer. We will talk a little bit about primers in a second, to allow this processing, and then remove this. And this is the lagging strand. So we have a leading strand that can go fast, and a lagging strand that requires different enzymes and the addition of these primers to allow the initiation of the replication. So we have a problem also in directionality, right? So what are these primers? DNA cannot be generated from nothing, from the polymerases that we know. We need an already known piece of DNA. That from there we can go. So we put something here that hybridizes, makes base pairing with what we already have, this orange red thing. And then from here the enzymes know how to run. So we have to have also these enzymes, the primases that form these little primers, so we can start the whole process. So for example, also when the genome sequencing project began to sequence all the organisms in the universe, what kept the research going slow was that we needed to go sequence a part, get to this end, then read it, and then generate primers that come from here to the next part. And this was solved by Sanger. He also won a Nobel Prize by saying, okay, let's add a step where we add random, when we take the DNA, we fragmented it in many, many pieces, and we stick it together inside a plasmid, right? And then what is a plasmid? You know what a plasmid is? Yes, some of you yes, some of you not. Okay, so plasmids are these basic units of DNA that usually bacteria have, and they can transfer from one to the other. And there are small units of DNA that can self-replicate. They have the information to replicate. But we can open them synthetically and then add this sequence from an organism. But we know where we open it. So we know the sequences in here, and then we can allow the sequences of all the genome in a much faster way. So by breaking the DNA in many, many pieces, putting in these plasmids, then what we needed to do is to align it, right? Because we have many DNA pieces. We stick them inside this plasmid. This is the sequence we are interested. But we already know this sequence because we put them inside. So we can read this part. But now we have to have overlap between these sequences in order to make them work together. And for this also we needed not only the biological technology, but also the computational capacity to align all of these sequences. And we have, again, many repetitive sequences in the genome. So just until last year, I think, was published a paper where we have a whole genome with the dark regions. What were these dark regions? Were all these repetitive sequences that we have that were very difficult to align. And these sequences include, for example, we have many, many viral sequences that we have been carrying for many, many generations, and they appear everywhere. So it's very difficult to map if they are in chromosome 3 or in chromosome 17, right? Because they look exactly or very similar to each other. So by doing this overlapping was not good enough. So new, also artificial intelligence and sequencing methods that go deeper, they finally managed to go and do all the sequencing of the genome. So we have overcome this dark matter inside our genome in recent years. Are there any questions up till now? Okay. So one of the things that happens is that we have replication errors, right? So it is good and it is bad. It is good because without replication error and without mutations, we will still be a single-cell organism, right? There will be no evolution at all. On the second hand, these mutations can be deleterious. They can cause disease or reduce the fitness. So what happens in a population is that if we have mutations that give us a better fitness, these are the ones that will reproduce and amplify. So we will have more of the ones that have better fitness. And those mutations that reduce the fitness will disappear over time. This is in the simplest explanation. And of course, for these mutations to pass on, they have to happen in the germline, or in the egg, or in the sperm. Because if it happened in our skin, they won't go to the next generation, right? So to have a mutation that goes on, it has to happen in the germline. And somatic mutations is usually what cause also variability. We will see that our brain and many different tissues have different genome sequences. We always generalize and say, okay, all of our cells have the same genome, but this is not true. First of all, there are some cells that go through recombination, like the immune system, because we want to generate all the different antibodies to prevent infection. So in there, we have genetic recombination of fragments to have a complete different genome of a certain region. But also we know now that one of the ways to gain fitness, for example, in the brain, is to allow retrotransposons, you know what retrotransposons are? To start moving when the brain is starting cell division before they differentiate. And in this way, we have neurons that are different from each other. And only the ones that are the fittest survive, but this gives our brain the capacity to have neurons with different capabilities, with different activations or mutations or this kind of thing. And also this could be bad in the long term, and we will talk about it in the aging class, but they are also important for development. So we have two types of cell division, mitosis and meiosis. In mitosis, we have our same cell divide and get the copies from our mother and father, so we didn't change anything. In meiosis, we go to an extra replication stage where we get half of the genome, and this, of course, only happens in germ cells, in sperm and ovocytes, to give us rise to organisms that depend on both parents. So again, I like to put you some movies at the beginning. After that, next class, I don't think we have more. Just in the introduction, I hope this helps.
A mutation is a change in the genetic code. These changes are often copying errors that happen during DNA replication. Some, and actually most mutations, have no effect on the organism and go unnoticed. Some mutations alter important genes, harming the organism and decreasing chances of survival. But some mutations alter a gene that gives rise to a new trait that actually gives the organism an advantage. Mutations are the ultimate source of variations in life on Earth, the good, the bad, and the ugly. Mutations are the reason for the variety of genes and traits that exist in our global gene pool. How can mutations lead to evolution by natural selection? Well, let's first talk about mutations that cannot lead to evolution. Some mutations only affect the individual carrying the mutation. These are mutations that occur in body cells. Mutations in body cells that happen during an organism's lifetime can only be passed on through mitosis to new body cells. The mutation will only be present in the cells that develop from the cell with the original mutation. Such an acquired mutation has no effect on evolution, even if the mutation is beneficial. Body cell mutations cannot be passed on to offspring. The only way for a mutation to influence the gene pool is if it can be passed on to the next generation. For this to happen in sexual reproduction, the mutation must be present in the gametes. Gametes are sex cells that unite to pass on genetic material from parents to offspring. Mutations in gametes are inherited by the offspring's first cell through fertilization. As every cell originates from the zygote, this mutation would be present in all cells of the offspring. If the mutation gives the organism an advantage and they survive and reproduce successfully, they may pass on this gene to their offspring as well. Beneficial mutations through natural selection may increase in frequency from one generation to the next. Evolution is the change of a gene pool over time, thus affecting the traits and survival of individuals in a population.
So again, we can use these mutations also to learn about evolution, because now we can know more or less the rate of mutations of different polymerases, and then we can use this also to see the differences between different species, how far apart are from each other, and also more or less the evolutionary time that it took to get there, right? Because if we know the rate of mutation that are acquired in a general way, sometimes there are hotspots of mutations, then we can also use this to understand about evolution and some traits that disappear or some traits that appear. For example, there is a lot of research right now trying to understand speech. And some of the mutations that seems to happen is that a loss of function and not a gain of function from monkeys to humans, where we lost the capacity to contract some of the muscles that allow for more kinds of vibrations that give us the ability to speak. So also there is a nice research from a mouse that discovered a gene that is in charge of the tail that was lost from a mouse to human. And when they mutate this in the mouse, they lose the tail. So there are many different things that we can investigate when we see loss of a gene or addition of a gene that can allow us to understand better evolution. And there are, for those that are more interested in going to evolution, there are so many websites where you can look for a gene and see how much it is conserved. And not only how much it is conserved, but which are the genes that have the same trend. Because we think if they evolve together, they work together. So sometimes we can have a gene that we have no idea what it does. We put in this context, so we see, okay, it started appearing only on mammals, and this is the same trend that gene A, B, C, and D have, and luckily we know the function of gene D. So maybe these are connected. So this evolutionary analysis can allow us also to understand novel gene functions and also how we evolve from different animals and acquire some characteristics. And this also helps us to study human migration or animal migration, right? We can see out of Africa and the different paths that different genomes took. If we look at the genomes or also the mitochondrial genome. So why is this again here? So this is also one of the tools that we can use by having genetic information to study migration and to study anthropology and archaeology and many other things that are part of what biology and research supports nowadays. And now if we go back to the germline versus somatic line, we go to the somatic mutations. And somatic mutations are the main cause for cancer, right? Because if we have a normal cell that has a first mutation, usually we don't get cancer. Our cells have to deal with it, and we have many systems that overlap to prevent the cells from losing control on cell division and reality. Usually we have a second and a third mutation, and these are what drive the cells to be malignant and become cancerous. So usually, if we are already born with a mutation that is bad for us, for example, in DNA repair, no, then we accumulate mutation faster if we don't manage to repair our DNA. And then the second mutation will accumulate faster. So these are the cases usually we see with childhood cancer, right? It's not, usually they are already born with some mutation. But for the rest of the population, our chances to get cancer increase as we age. Why? Because as we live longer, we accumulate more of these mutations, and the chances of getting double and triple hit in something that is important increase as we live longer, just in a statistical manner. So this is also what is the main cause of cancer, and they go through changes to adapt to new metabolic challenges, to hypoxia. For example, if the cells start growing, then they have less oxygen, but then they have some ways to overcome this because they are constantly changing, and only the ones that survive keep going. So we can also use mutation analysis in cancer to understand how or where did it started, and try to predict where it is going, because one of the main problems we have nowadays in cancer research is that the medicine, all the chemotherapeutic or different things that we use to prevent cancer induce damage, and many cells escape by mutating even more, and then they become resistant. So many times we see that cancer appears again a few years later, because maybe we didn't get rid of all of the cells, and maybe some of the cells also were resistant in the first place. And this is very similar to some research that was done in bacteria. They saw that all the bacteria is growing at a more or less steady state, but there are some bacteria that they are very, very slow. They divide very, very slowly, and you can see this in the film. Then you wash them with antibiotics, and all of the fast-growing cells that are the main population, because they divide fast, they form more of themselves, die, but these slow-growing cells are the ones that keep alive after antibiotic usage, and then they start reproducing again and start having again the population that divides faster and divides slower. So we think something very similar happens also in cancer. We have fast dividing cells that form the tumors and migrate, but also we have some slow dividing cells that they become more resistant and are more difficult to get rid of. So this is why it's also important to try to predict the trajectory of novel mutations that these cancers will acquire, and what is already mutated to try to target with personalized medicine, because this is another problem that we have, that we use compounds that kill all the dividing cells. So that's why the hair loss and the cells stop dividing, and many side effects. But if we know what to target, we can reduce the dosage, and we can prevent the side effects. So this is also one of the things that is important in genetics that we are using as a tool for personalized medicine. So one of the examples that we have with cancer is Bracha mutation. Bracha mutation is a very relatively common mutation that leads to breast cancer, and carriers of these mutations have a 50 to 65% chance of getting breast cancer. So there was a very famous case with Angelina Jolie, right, that she discovered that she has a mutation and decided to remove the organs to prevent cancer. And this brought a lot of awareness to Bracha mutation. We will study more about Bracha in the DNA repair class, because BRCA1 and BRCA2 are enzymes that help repair the DNA. So that's why if we don't have them, we have already more chances of gaining the novel mutations and having DNA damage that will lead to high risk of cancer. Okay, so now we will skip to other functions of the cell, but for the following days we will go back to the genome and the epigenome, and we'll just remind everyone that is not a biologist about the other things that happen in a cell. So we have the central dogma, that of course is not true, that from that we go from DNA to protein and never back, right? So we have our DNA, we copy to prepare an RNA molecule. This RNA molecule gives us the information to form a protein, and the proteins have function in the cells. Of course, we know now that mRNA also has functions, that we have also long non-coding RNA, siRNA, shRNA. We have so many other types of function, and also that some cases we can insert RNA into the genome and generate new genes. We have reverse transcriptases inside our genome that can put this back. But for many years, it was thought that this is the only direction that can be done. And again, what we know about the gene is that we have our DNA, they have exon and introns. So for those of you that didn't study that, the intron is a part that is non-coding, and the exon is a part that it will be part of the final message. The RNA copies everything, and then we go through the splicing process where we remove the introns, and then we get the final phrase that we need to translate. But we have one gene and many different messages. Because we can switch the promoter, we can use this one or this one, and then we have different mRNA that is just change a little bit or just change when it will be used. Because if I use a different promoter, it can be activated maybe by heat shock, and another time it can be activated by an hormone, right? If we use a different promoter, the same gene can have a different activation. We can have cassette exons, meaning that we can put it in or we can leave it out. Again, this may change the whole meaning of the phrase. If we think about a phrase, we can add the word no. It will completely change what it says. So it's the same with genes. We have mutually exclusive exons. If we have this, we will never have this, and vice versa. We have exon scrambling, that they can be, and this is very rare, but they can be accommodated in different ways. Alternative 5' splice sites, so we can have this inside or not, but this is not in the middle of the genome. It's not an intron, but it's part of this exon that can be get in or out. The same on the other side. Some cases we have retained introns, and this is less common and is more seen in disease. And alternative polyadenylation, meaning that the end of the gene can have a different poly A signal that is also will dictate how long this mRNA can live, right? How long do we want it in the system? So this is one of the most common examples, because it's amazing how from a single gene in Drosophila that is called this CAM, is expressed mainly in neurons, we can have 38,016 different mRNAs by a combinatorial of these different cassettes. So we will have only one of these that can come with one of these, that can come with one of these, and here we have two alternative exons. So if we do the combinatorial of how many different transcripts we can have about 38,000 different transcripts from a single gene. So again, when the genome was discovered, everybody was expecting 150,000 genes and humans to have so many genes, and in the end we have like 30,000. And it was kind of disappointing for everyone. It's like, how come we have so little genes? But then if we add all these layers of regulation, then we have one gene that can be 38,000 different things. Okay? Epigenetics means literally above the genome, because when we change the epigenetic code, we don't change the genetic code. It remains as it is. But we change how it will be used. And this information can be passed from one cell to the other when they are dividing. And sometimes, for example, in C. elegans, we can have also a transgenerational effect of epigenetics. In us, for example, what we know until today is that we have to erase all of the modifications when the zygote is formed, most of them, because we have imprinting. That some regions remember if they came from the father or the mother. We will talk about this as well. But most of the other regions are deleted in order to create all of the cells from scratch again. In C. elegans, through siRNA, they can remember things that happened to their great-grandfathers. So it's a little bit different, and people are trying to figure out if we also have this capacity, but for now we don't know yet. And many times this can also lead to disease. Many times we don't find a mutation in a person. We see that there is nothing new, no somatic mutation, but the epigenetic change is the epigenetic code is changed. And then we are using the genome differently. And this makes it a waste of energy and sometimes to express genes that should be silenced and should not be expressed. So this also can lead to disease. So if we, again, in broad terms, say that every cell has the same genetic code, but every differentiated cell has a different epigenetic code, then we can control with this variability, with this combinatorica, what is the outcome of the cell. And we will go through that later on in our class about stem cells and IPS, induced pluripotent stem cells. So now we will go back to our cell and go to the endoplasmic reticulum. So the endoplasmic reticulum is, the membrane is adjacent to the nuclear membrane. It's the same membrane that continues. And when the cell is dividing, this nuclear membrane retracts into the ER. And when the new cell is formed, it comes again out from the ER. Because if you remember, I said we never form a membrane from zero, from scratch. And the nuclear membrane, after in the cell division is gone, it comes back from the ER. And it's used for protein folding and maturation, lipid synthesis. We need specialized domain to generate lipids because it's not comfortable to have them in the cytoplasm, right? It says repel liquids, so we need a special place. Calcium storage, and all our muscles depend on the release of calcium and internalization of calcium again to generate movement, and formation of membranes. So if we have proteins that they need to be formed and arrive to different places, many of them will be translated in the ER and then sent wherever they need to be in the cell, modified with glycosylation and other modification, and this happens in the ER. Other thing that happens in the ER is lipid biosynthesis. We generate cholesterol and many other different lipids inside the ER. And we have the calcium storage that we were talking about, that is released as a signal. And this is very, very short movie, but it's really, really cool. These are calcium sensors. And when the calcium is released, you can see a pulse. So these are neuron-like cells. And they have a calcium sensor that when we have high concentration of calcium, you can see red. And low concentrations of calcium, you see more bluish color. So we can track how cells are activated and pass the signal with these calcium sensors. Okay. And we have the main activity that is known for the ER is the synthesis of protein folding and structure maturation, and addition of glycosylation and disulfide bonds and many things. So all of this is happening in the ER, but also the ER is very important for the maturation of tissue. For example, all the tissue that secrete things. For example, if we have the immune system, we have to generate antibodies. We have to have an enlarged ER. If we have problems in this, the cells will die and we will lose capacity. The same is true for pancreas, for the B cells. In the brain, we also secrete a lot of neuropeptides. So problems in the ER, mutations that cause problems in the ER or unfolded protein response will lead to diseases that we see a lot of metabolic disease, immune system disease, and neurodegenerative phenotypes. Okay, and the ribosome, that is the basic unit that attached to the ER to translate the proteins, the structure was discovered by Ada Yonat, and she won the Nobel Prize in 2009. She's in the Weizmann Institute in Israel, and she worked for it like 20 years, but of course this is schematic, but she discovered how this ribosome works and translate the RNA with the two subunits, the small subunit and the large subunit. And we won't get inside of the ribosome because our focus is somewhere else, but just to say that it's a huge molecule with many, many different proteins, RNAs, and a very complex machinery. But also, since it's a very complex machinery, it's evolutionary conserved from archaea and from many other organisms. So in many cases, when we don't have information, for example, when we go to deep sea water or we go to anaerobic systems that we cannot grow these animals, we have no way of the conditions to know how to grow them, what we do is we sequence the rRNA, the ribosomal RNA. Because it's one of the most conserved genes, it's present everywhere, And through this, many of the ecological microbiome or ecology systems, this is what they use, ribosomal RNA, to try to understand how the species separated from one each other, and also how many species do we have. We take water from the deep sea, and we don't know what is in there. But by sequencing the rRNA, we can get insight of how many species are there. This is how the ER looks inside the cell. So it's a very, very big network. And I am not sure these statistics are correct, but they say that under 20% of the proteins are correctly folded, and the others are sent to degradation to try again, because folding the proteins is also a very complicated task with many different possibilities of folding, right? We have a linear peptide, but when we start folding it, we can do many different things and get a non-functional protein. So the proteins that are not folded correctly, they can also get help from chaperones, that they help them fold correctly. And if they are not folded correctly, then we get to degrade these proteins. So many diseases, mainly also neurodegeneration, but also in vascular disease, we have accumulation of aggregates. Like in Alzheimer, we have amyloid beta and tau and alpha-synuclein, and all of these aggregates, at the end of the day, also cause the cells to die. They are toxic. In the bloodstream, they can accumulate and block the passage of blood and oxygen, and then we can have a stroke or we can have heart problems. So non-folded proteins that accumulate are a big problem for us, and they have to be dealt with. The second organelle we'll talk about is the Golgi Vesicle. It's mainly the mailing system of the cell. So after the cells were prepared in the ER, they pass to the Golgi, where it is decided where they have to go in the cell. If they go to a membrane, if they go to a different organelle, if they should go somewhere specifically in the cell. So this is the mailing system, and some other processes happen there, but this is really an under-investigated organelle in the cell, and not a lot is known about in recent research about it. But it regulates the secretion of different proteins, molecules, also sometimes the internalization. They can go back and generate a stress response. Then lysosomes in the cell are in charge of degrading and recycling materials. So many times we use the proteasome to degrade small proteins, but if we have to degrade a full mitochondria, or we are in a hurry to get energy because we don't have enough energy, we are under stress, we can degrade this with lysosomes and autophagy, the different materials that we need in the cell. So we have the lysosome, and I don't know why it's going so slow. So they are membrane-limited organelles, and they go and acidify the interior. So the proteins are degraded, or organelles degraded with these hydrolytic enzymes. And we have the proteasome degradation, but this is a more targeted way to destroy a protein. So why do we need to degrade proteins? So first of all, quality control. As I said before, if we have unfolded proteins that form aggregates, they are toxic, and we have to get rid of them. And also, they are not doing their job. So also, we need to eliminate proteins that are not needed right now. And we will go through some examples. For example, in the circadian clock or a cell cycle, we need the protein, but then we have to get rid of it because we don't need it anymore. We need sometimes recycling material if we are under starvation or if we have other problems. And the antigen presenting system, that we also give a few examples, how it helps us to recognize antigens and bacteria and viruses that come to the cells. So quality control, if a protein is damaged, as we said before, this is the folding scale of what are the different energetic values a single protein can have to get to the correct folding. So if we get to the incorrect folding, we want to degrade this protein. And if we get to the correct folding, it can have partners that prevent its degradation. So we have many different outcomes, and if we don't get the correct one, we want to get rid of this protein. And in here are some of the examples I was telling you about. Amyloid beta and tau, alpha-synuclein proteins, and transgeneration can all form aggregates that are toxic for different systems, but we mainly know many toxic proteins that affect the brain. then we need to eliminate proteins that we don't need right now. So if we imagine the cell cycle, and this again is just very short to show you a cell dividing. Okay. It was not important, but it's super cool to see the cells dividing. Maybe later I'll upload the pictures. So one of the systems that the cell uses to divide is to prepare proteins that are cycling through the cell, through all the phases, and degrade them at some point. So for some phase, we need a lot of cyclin E. But then when we move to the next stage, we have to get rid of this cyclin E. So of course, we can regulate this also at the mRNA level, but also at the degradation level. So we use it, we don't need it anymore, we add ubiquitin, then we send it to degradation. So for example, we have these cyclin proteins that attach to CDK, and we need, when they are attached, they are prevented from going through degradation, but then they start getting modifications, phosphorylations, and ubiquitination, and get degraded. So the CDK is free again to take a different cyclin protein. So in this case, we can degrade the proteins that we don't need. Also, another thing that goes cyclic in our body is the circadian clock. How are we awake or go to sleep? And for this, we also use mRNA that goes cyclic, but also proteins that accumulate and can start the degradation process through modifications to help us understand that a cycle has ended and start a new cycle. So this helps us understand where are we in the day. Sometimes we have stoichiometric excess, meaning we produce too much of a protein and it doesn't have its proper partner. So for example, if we have a lot of this protein with the degradation signal, but it finds its partner that is a protein B, it protects it from, it moved too much, it was supposed to cover this, but don't worry, that's the idea. So these other two proteins will be degraded because the degradation signal was available. So this controls the formation of complexes in the cell. And we have the recycling materials. If we are under stress or we want to use them again, we degrade these proteins, and we can use it also for new amino acids, for new proteins. We can use it for nucleotide synthesis. We can degrade the proteins and use it for nucleotides, and also for energy, for acetyl-CoA and the TCA cycle. So we can use all of these amino acids for different things in the cell. And last is the antigen presenting system. That is a very interesting thing. There are many examples like this, but I present only this one. So we are constantly putting in our membranes these receptors that they present some of our proteins that we have inside the cell. So we degrade some of the proteins on purpose and take these peptides and present them in the membrane. So when these proteins are cells, they don't activate an immune response. It goes, checks, and it's like, this is yours. We know this one, it's okay. But if we get infected by a virus, a bacteria, or something, and we are producing these proteins that are not ours, some of these peptides will reach the membrane. And then if the immune system comes and sees, it says, okay, this is not a self-protein, we will activate the immune response and start killing the cells. So we degrade proteins on purpose to also use them and present them in the membrane to activate the immune response and protect ourselves. So how long proteins live? We can have from minutes to days and even months. And it depends on the functional and genetic sequences and the quality of these proteins. And how we degrade them, we put the death mark in them. That is the ubiquitin molecule, right? So when we don't want this protein anymore, we add a ubiquitin molecule that was discovered by Chehanover, Hershko, and Rose, and they got a Nobel Prize in 2004 by this ubiquitin system. So what happens is that if we have a protein that we want to degrade, it becomes, for example, misfolded, or a phosphorylation or something else allows the exposure of this site. And then we have to add polyubiquitination, because ubiquitin can also be used as signaling molecule, but if we have many, many ubiquitins, then it will be sent to the proteasome for protein degradation. And the proteasome looks like this barrel, where at the beginning we have some helicases, like protein helicases that help unfolding the protein. So it goes as a single amino acid line that is cleaved by 6 different proteases that are located in here. And then we have these small peptides that are released and go outside of the proteasome. Okay, so this is for the proteasome. Then the lysosome, we talk about it a little bit. And we'll go to the mitochondria. So the mitochondria, everybody have heard many times, is the power cell of the powerhouse of the cell. It helps to produce ATP, and it was engulfed many, I don't know, millions and thousands of million years ago by an archaea cell. And we are using, usually the mitochondria had many more genes, but most of these genes were transferred to the nucleus. So now there are only 16 genes in the mitochondria, and about 1,000 genes that are expressed in the nucleus that go transported inside the mitochondria. And in this way, we keep the system together, and they depend on each other. We cannot have no mitochondria. The mitochondria always, again, comes from the mother. And in the mitochondria, we have what we call the cellular respiration. So we always know that breathing, we get O2 and release CO2, but all this goes through a process inside the mitochondria to produce energy, and the last acceptor of this molecule is the oxygen, is produced water. So we need the oxygen to accept these electrons and release. And for example, cyanide or these venoms, what they do, they block this chain, and then we may get oxygen, but it's stuck. So we will see that we have different complexes that pass the electrons from one to each other to the final molecule that is oxygen to produce water. And in the way, we move protons along the membrane of the mitochondria to form the mitochondrial membrane potential. And this potential gives the strength to this beautiful molecular machinery that forms the ATP. So again, I'll show you a movie because it's really easier in this way to see the machinery. It started? No.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double membrane organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers.
Okay. It's not very important for me that you remember all of these things, but mainly to bring you like the movie, because it's really a beautiful molecular machinery. And it just gives you the idea again that we have the energy that is coming from passing electrons from high energy to lower energy until the acceptor, that is oxygen, and that's why we breathe. And using this energy to bring protons to the other side of the membrane, and then using this as a turbine to generate energy from this cyclic movement. So we will skip this part, and we'll go to the last part, and this is how the cells die. And I promise that if you want the movies, I send a PDF with all the presentations. I hope you already can see them, but the movies I will upload later because you cannot see them in the PDF. But the last part is like, okay, we generated the cells, and now they have to die at some point. So they can die by injury, mechanical damage of the exposure toxins and in an organized way, or by suicide. And we get external signals or internal signals, depending what is the cause of death. So mainly there are 2 mechanisms, apoptosis and necrosis, but we know today that we have in between so many other necroptosis, ferroptosis, many other pathways, but we'll main in the 2 more known pathways. And in apoptosis, what happens is that the cell understands that it has to die. It gets signals and starts shrinking. The organelles are reduced. The mitochondria, that is very important, start leaking cytochrome C. The leakage of cytochrome C activates the apoptosome. And then this is irreversible already. We have chromatin condensation and nuclear fragmentation. We send enzymes to cut and chop the DNA so we can pack in small membranes. We have this beveling, and this is packed for the immune system to eat and not have the waste going around. So when cell is apoptotic, it starts releasing, it changes some of the membrane lipids are switched, are flipped from one side to the other. So now they are recognized. We have the B layer is not symmetrical. We have different lipids outside and inside. So the ones on the inside, when they are flipped outside, they are a signal for the immune system to recognize them and engulf them. So when we start packing everything, we form these small mini cells containing the DNA and the organelles, and the immune system can engulf them and get rid of them. Any question? No? So why should a cell commit suicide? Of course, they can die by necrosis, but it's an aggressive cell death, so it happened and they don't have control over it. But we can have apoptosis for very good reasons as well. For example, for tissue formation, like breast tissue after lactation. So we have a function. We have to feed a baby. We have to enlarge the breast and produce milk. But after this function is gone, we have to kill the cells. We don't need them anymore. So these cells can go through apoptosis. And we have also the formation of some of the fingers. At the beginning, everything is continuous. But then we kill these cells in the uterus and in the brain. So we have some examples, for example, in the immune system, when we are developing, we have all the combinatorica that was produced randomly by DNA recombination. But some of these antibodies may recognize cells. And then we can have an autoimmune disease, right? So we want to get rid of all of the cells that recognize self. So during embryonic development, if the immune system recognizes self, they will kill themselves. After development, after we are mature, is the opposite. Cells that recognize some antigen, they will start cloning themselves and produce antibodies. But during development, we want to kill these cells because we don't want an autoimmune disease. The organ size, if we want to limit the size of the tissue, these are, for example, the fingers in the hand. So at the beginning, all of this is produced, but then we start killing these cells to have our limbs. For example, in the brain, we have many more neurons than what we need during development, but only the ones that reach the signal or another partner cell will survive, and the other ones will commit suicide. So we have an appropriate number of cells in the brain. And this is, for example, as we said, in lactation, or if we remove testosterone, for example, chemical castration or development, then the prostate gland will die by apoptosis. So we need also apoptosis to get rid of infection. If we were infected by a virus, it uses our machinery to divide and infect many other cells. So it's better if we commit suicide in an altruistic way, right, to prevent the spreading of the virus. So this can happen also by other systems, or if we have DNA damage and we know that we'll accumulate mutations, then we should commit suicide to prevent this and produce cancer cells, for example. Or we have two choices. We can become senescent or we can become apoptotic, and we will see this in another class. But in some cases, if we have too much apoptosis, we start losing tissue capacity. But in other cases, if we don't kill these cells, we have cancer, we have hyperplasia. So it's very important to keep this balance between cell death and keeping the cells alive. So these are, again, are the different ways that we can kill a cell with different signaling for virus infection. And usually we have in the cells already pro-apoptotic proteins and anti-apoptotic proteins that are expressed all the time. And they are in certain balance, like blocking each other. For example, BAX can prevent the function of BCL protein. But if we have a difference in the stoichiometric production of any of these, then we can switch the balance for apoptosis or survival, depending which one of the proteins is produced more. And if we have too much, again, we have tissue atrophy, neurodegeneration, a thinner skin, and many of the things that we see also in aging. And if we have too little, we can have hyperplasia, including cancer, atherosclerosis, and more. And that's it. So we went today through evolution, through the cell and all the organelles. So next class, we will go deep inside the nucleus and the epigenetic code. And that's it. So if you have any question, also ask in the middle of the class. It's okay. And I don't know, any questions for today? Okay, so you are free. I see you have a... there was a question? Oh, yeah, sure.
It came up to me during the presentation, and now you said it again. So what do you mean precisely by epigenetic code? Because I tend to think of genetic code as the rules or a table to translate codons of DNA to amino acids in a protein, or from codons of RNA to amino acids in a protein. And I do not understand precisely what would be epigenetic code.
The epigenetic code.
Yeah.
Okay, we will go to a full class about that, but let me tell you in a very simple way that when I have it, if we have the genetic code like a book of instructions, with all of the instructions, like a script for a theater, right? And then each actor has to read its own part. The other parts are not important for him, so he can mark which parts are the ones that he has to say. So this is part of what the epigenetic code does, right? I mark the parts that are important for me as a neuron cell, and a kidney cell marks the genes that are important for him, and all the others are not important. So these modifications that you do with the yellow marker is part of the epigenetic code. Also, the epigenetic control can help control how to condense the chromatin or open it, because you have to pack about 3 meters of DNA in each one of the cells. So you have to pack it correctly to allow the proper usage, which parts are open, which parts are closed, which parts are dangerous. And all of this will be regulated not at the genetic sequence like ATCG, what it means, but how I fold it and pack it, and which flags I put, like this one means read me, this other one means don't read me, keep me close, whatever you do. This means put me together, chromosome 1 with chromosome 3. Let's stick together because we have to go regulation together. So the epigenetic code is this modification each actor can do on the script to benefit himself, and it can also respond to the environment. For example, if we saw that the play was a disaster and no one understood it, maybe I have to do it with more passion, or I have to do it slower. So the epigenetic code allows the adaptation to the external signals. So imagine it as a script. Not everybody in the play has the same genetic code, the script, but each one has marked something different to use it differently. And we will go through all the epigenetic code in the next class, okay? There are more questions? No? Okay, great. So you are free.
Hi, I'm again here. Just a quick message. The coffee break this week at least will be here, as you can see, right? And you have a break. You will have the next class will be only during the afternoon, okay? So you have a break. During this break, please fill the form evaluating us, evaluating the courses of the introductory module, and write answers there, right, guys? Not like put just the numbers, right? Make evaluations, good evaluate, not good, as you think, but make a good quality evaluation, please. Yes? I cannot listen you. In English, please. Yeah, for some of the professors, we are going to send evaluation. We don't have access to your names. We don't have access to your emails. It's just so people know how they were like teaching you, where they can improve. Okay? Thank you.